right. So can you all see the screen, the slides?
Yes,
Yeah,
Yes.
all right,
Yes.
coming through.
great. So I'm quite happy to talk about the result of this year and show what we did in terms of assay, in terms of trying to reduce irrigation in nursery, trying to reduce water use, trying to find different way of doing that. So we're going to show a little bit about the project, the objective, and what we did and what we achieved. So as you all probably know, irrigation in nurseries is essential for plant quality. So if you don't water the plant enough, you don't have the full growth. And if you water the plant too much, then you have trouble for the roots, so not enough oxygen, or you're just losing water, which can be quite a problem in some nursery because you don't have that much water available for the industry. So we are trying to find a way to kind of reduce the amount of water that will be put on the plant and still get good growth in terms of the plant itself. So irrigation results often in significant water loss. Overwatering and leaching is a problem, water not reaching the pots also, and evapotranspiration. So just the water on the surface of the leaf can not reach the roots and you would just lose that also. So we have three different objectives for the project. The first one was to optimize irrigation management using wireless tensiometers. This was mostly what I showed you last year. And we also did continue a little bit on that subject this year. So I will tell you more about that later. One of the goals of the project is to establish the best clustering practices. So we did start that in the first cluster we did a few years ago, and we had really good result, but we want to expand that to get even more perennials and shrubs to get really have a good guide for the growers in terms of how to put plants together for their water needs in itself. And the third one is to compare different strategy for the automation of irrigation. And that's really one of the things that we want in that project, is to try to make it way more automatic, so we don't have someone that is looking at it several times a day. We want to have some kind of way to measure it and also start irrigation automatically if possible. So we're going to talk a little bit more later about that, but let's go back to the clustering practices. So the clustering that we did in the last project was based on 10 species, and the goal was to cover a wide range of water needs. So we had like plants that don't use a lot of water during the season, like for example, Asta is not growing really quickly, so it doesn't use that much water. And at the other hand, we had shrubs like Physocarpus that was using a lot of water. But we wanted to have new species to do the clustering, because we thought we had some places where we didn't have enough range in terms of water needs, especially for the species that use a lot of water. We thought that we could get maybe one or two more species that would be good for that, and also species that can grow really quickly in the season, because as you will see later, some species will grow really quickly and need a lot of water fast and then slow down their growth. So we wanted to have more diversity in terms of species that we want to use as a reference. So to do that, this year we use 4 different species: Sembicus canadiensis, Salix integra, Rudbeckia fulgida, and Panicum virgatum. So those were actually we thought would be good candidate to kind of increase our range of species. We did the experiments at Laval University Nursery. So it was under a tunnel, so we didn't have any rain. So it was a good way of really measuring everything. And as you can see, it was quite a complex, you can see all the solar panel. So we had wireless tensiometers that were connected to solar panel and were transmitting all the data by cell signal. We also had, all the plants were in 2-gallon pots with bark-based soil mix. It was agromix N7, which is a mix that is often used in nursery. So it was a good standard to use. And it's also a substrate that is really forgiving for the shrubs and the perennials. So it's a really good substrate to use. And we also had automatic valves connected to the tensiometer hub. So basically, as soon as you had the tension that you wanted to have, so the soil was at the right water content, you could start the irrigation without having anyone having to press or going to open the valve like the first cluster we did. We also had water meters, micro sprinklers, so all the plants were irrigated one by one. And so that's it. That's pretty much the experimental setup. So let's go to the result. Oh, sorry. Before that, just to illustrate exactly what are the tensiometer. So you can see in the middle, this is the probe that we're going to put in the soil. So it's a ceramic probe, and the water is getting in and out. So that's how you can measure actually if the water is available to the plant or not. So it really kind of represents if the water can be taken by the roots or not. So you can see here, it was put in 2-gallon pots, and you had here the hub, the tensiometer hub that was connected with 8 different, either the valve or the tensiometer in itself. So that was the setup in terms of a wireless tensiometer. And you can have actually the data from anywhere, wherever you are. So it's either on your cell phone, on your computer, you can see right away what's going on everywhere. So it was really, it's really efficient if you have a lot of different plants and things like that. You can see all of that at the same time. So we had, what we wanted to do with those model species was to determine what was the best condition in terms of root growth, what do they need? And we had 3 different thresholds. So the first one was kind of the wet one. So basically when you reach -4 kilopascal, then the threshold is reached and you water until it's completely wet again. So it's basically always stay moist. And then you had the average treatment, which is here in yellow, where you could go up to -8, and the drier treatment, which would go up to -12. So -12 when you touch the surface of the soil is quite dry, but it's not to a point where the plant will wilt and be completely dry. So it's still in a range where normally the growth is able to be done. But we wanted to see which was the best condition for each plant. And normally what you would see is if you have a drier treatment, you would use maybe a little bit less water, not that much, because often to go from -4 to -12 when the plant is quite big, it's a matter of minutes and hours. So it's quite quick, the cycle, when you get to those drier part. But still, it's often you're able to save a little bit of water there. So we wanted to see what was the best solution there for each plant. So let's start with the Rudbeckia. In terms of growth, we didn't see any big difference between -4, -8, or -12. So it was really kind of similar in terms of what they need, or what they, in terms of growth at the end of the season. And it was pretty similar also for the water use. So we had a slight decrease at -12 and -8, but not by a lot. So basically it was just at this point, because the growth was kind of similar, the water use also, it was just to look at the appearance of the plant. And what we saw was that the -8 was a little bit better, than the -12. And even the -4, because the -12 was still good. You see the leaves are well and the plant is growing well, but there were some kind of delay in the flowering. So if you want to sell quickly your plants, you're better to go with the -8 for Rudbeckia. It's not something that we see in all the species, that kind of delays in flowering. It's one of the only one that we really saw a big difference in terms of delay of flower. But because of that, we decided to go with the -8 there. For the Salix, this one was really quite obvious that we had better growth at -4. So it's a plant that really liked water. You can see the roots are just trying to get under the pots and trying to go even under the plastic that we did put underneath the plastic cover. So it's a plant that likes water, that would grow way better in terms of water. You can see, however, that at -4, it's going to use a lot more water also. but the growth was so much better, even in terms of if you had like any kind of small damages for insect or things like that, it was growing way better right after. So we think it's a better choice to stay in the moist part in terms of growth for Salix. And you can see here, just with the pictures, you can see at -4, it's better than -12 in terms of general appearance. We didn't do any kind of cutting during the season because we really wanted to see the entire growth that you could get versus what you would do in a regular nursery setting. For the panic, it was it was the first grass that we use in terms of model species, and you can see that the growth is pretty much the same. Water use is similar also, but we had less variation with the -8 treatment. So less difference from pot to pot compared with -4, where we had big differences. And just the general appearance was better at -8, -12. Sometimes the leaves were rolling a little bit, so we didn't like that, and we advised to go with -8 for that species. And it's the same for the Sembicus canadiensis. In that case, we had some kind of something really kind of weird. So the growth is pretty much the same, so it's not surprising. But normally when you decrease the amount of, you get to the drier treatment, you normally reduce the amount of water, not by a lot, but often enough that you can measure it. But in that case, it was the other way around. I think it's mostly because the plant had trouble to keep all the water, so maybe they were more leaching. But we couldn't figure it out because it wasn't that obvious, and we didn't see any difference with the other species. But either way, that treatment is not the best because it's used more water and it's not bigger at the end. So -8 is the best option there. But as you can see, pretty much all the treatment are kind of similar. So the most important part is really how they going to be used as a model species in terms of clustering. And this is what is interesting and important here. As you can see, the Salix is, at the end of the season, the one that use by far the most water. So it's probably our species that will be the most useful to cluster with species that need a lot of water. And you have Rudbeckia and Sembicus that are kind of in the middle, and you have Panicum that is a little bit lower. And I told you about Asta before, and we also had Tsuya and all those other species that were way below here. So with all those 4 new ones, we have like complete range of water need. But the other thing that was really interesting for us was to see the difference between Salix and Rudbeckia, for example. So you can see here that Salix is using maybe 30% more water than Rudbeckia. But if you go after 1 month of growth, this is the other way around. So Rudbeckia is using a lot more water than Salix. So if you cluster them together, Rudbeckia will be lacking water at the starting of the season, but at the end will have too much water if you control the irrigation with Salix. So this is just an example how we can get a different kind of curve, and it's going to be a good model species for other kind of crops. So Rudbeckia and Sembicus are pretty much the same. They are more linear in terms of growth, and you have Salix that takes a little bit of time to start growing, and once it started, get really quickly grow. The growth is really getting much higher, and you need a lot more water also. So we were quite happy with those results. And we're going to be able to use those reference species next year and the year after. With the Salix will be used for high water need, and Rudbeckia and Sembicus for early growth, and Panicum will be a middle range. So we're going to be able to test 50 new species in association with those reference species. We're going to have perennials and a shrub, different than what we had in the first cluster. So we should have a total of more than 100 species that we're going to be able to see how they can cluster together. So the second part of the project for this year was the automation strategies. So the setup is similar to what we had at Laval University, but it was in a commercial nursery without any tunnels. So it was with the rain and everything. We had wireless tensiometer, automatic valve, a weather station. all of those were automatic, so we can control all of that. We had 2 species in cluster, the same bark-based soil mix, and we had water meter and the sprinklers. And you remember last year we had trouble with our sprinklers. It was not really good quality sprinklers, even if they are supposed to be good, they are not adapted for nursery. So this year we have way better model than last year. So we had more uniformity, and we wanted to see if we could improve our result with that, having less variation. So we have 4 treatments this year. So we have irrigation that start when the threshold is reached, a little bit like I just showed you with the model species. We had full irrigation, so you get dry and then you water until it's completely wet, and then you do those cycles. We also had the small interval, which is you always kind of stay moist, but you don't have soil that is too wet or too dry. So it's supposed to be the best in terms of growth of the root. The downside of that is that you're going to have really short cycles, so you're going to water really often. So maybe every 40 minutes or 1 hour, but only a few minutes every time. So what we saw last year, it was really efficient in terms of water use. So we use a lot less water when you do that. And it was really good with drop irrigation, but we wanted to see if we could do that with bigger sprinkler. And since last year we were not happy with our sprinkler, we were hoping it would work this year with better sprinkler. We also had irrigation based on prediction. So a prediction, we use evapotranspiration data, and we had 2 different coefficients, and I will come back to that later. So let's go back to the tensiometer treatment. So we had the full irrigation and the small interval, like we can see here. So this one always kind of stay in the best range in terms of root of growth, and this one will have more variation, but will also have more space in between irrigation, so it's easier to handle in terms of schedule of irrigation. For the evapotranspiration, we use the Penman-Montate equation that takes into consideration the temperature, the wind, the solar radiation, the relative humidity. So it really pretty much take everything into account in terms of environment. And then you adjust for the crop and the stage of the growth, because this equation is based on alfalfa in the field and some kind of height of the plant. So you need to adjust that to your plant. And the irrigation was made once a day based on the estimation of water lost during that day. So it's basically you lose water and you put it back every night with the estimation of what you lose. So this is the daily evapotranspiration at the commercial nursery. And as you can see, it's quite variable. So you have days where you lose a lot of water like that. And then the next day you can lose a lot less water. But as you can see, when you're in the peak of the season, so beginning of July and June, you lose a lot of water really quickly. And then it start to slow down when you get to August and September. So normally you need to apply less water at the end of the season. However, your plant is growing a lot, so you need to adjust that and have your coefficient based on the stage of the plant. So if we take, just to give you a small example, if your daily evapotranspiration is 7 millimeter based on the equation, then you need to adjust it after with the crop coefficient. So if you have, for example, hydrangea, and the coefficient is 1.2 in August, mid-August, then you just do 7 multiplied by 1.2, and then you say, okay, I lost 8.4 millimeter of water, and then you need to look at how much time do you need to start your sprinkler to have 8.4 millimeter of water. In our case, it would be around 20 minutes of irrigation with our sprinkler, but it varies depending on the sprinkler you have. So the most important when you deal with evapotranspiration is to get the good coefficient. So this is the crop coefficient that we had this year based on the hydrangea. We had tensiometer in the soil, so we're able to adjust the coefficient during the seasons. And as you can see, it's quite linear, but you have several steps. So those steps are often because you're going to have rain. And during those days where you have some rain, you you often don't need to water, so it's more difficult to adjust the coefficient every single day. But you can see that it's increasing during the season. The plant is bigger, it's going to need more water, so it's quite obvious. But it also shows that you need to adjust it quite frequently, so several times or twice a week maybe, to get the best coefficient in terms of efficiency. So let's see the results now. Did we have any differences in terms of growth or of water use? So let's start with the growth. We had two species in clustering. So you can see for the hydrangea or the Sembicus, it's basically the same thing. So you have no differences between your tensiometer treatment, so the full irrigation or the small interval, or between both coefficients, so the one that you put a little bit more water or less water. Pretty much it's the same thing. Same thing for Sembicus here. So basically, that's a good news. We're quite happy with that. You have no differences in terms of growth. So the plant is, whatever treatment you're going to use between those four, you're still going to get the good growth. So we wanted to know now, okay, do we have differences in terms of water use based on that? And
we have 5 minutes left, Charles.
All right, thank you.
Just a warning.
So in terms of water use, that's where we saw the most differences. So the full irrigation was the most efficient in terms of water use, used a third less than the evapotranspiration. So as you can see here, the full irrigation was the best treatment. The small interval, where we had really good result in terms of drip irrigation with sprinkler, doesn't work. Well, it's pretty much the same here, but you don't have a reduction in terms of water use like we saw with the drip irrigation. So it's not a technique that should be applied for that. And then evapotranspiration here, it takes a little bit more water, but still we are able to get good growth. So this is the takeaway message. So tensiometer are highly efficient. The choice of the pot with the tensiometer is crucial. So you need to avoid the smaller plants, take the bigger plants, or the plants that are more representative of the population. Full irrigation is better than small amplitude with sprinkler. It wasn't the other way around with drip irrigation. So it really depends on the system that you have. And in terms of evapotranspiration, it's less expensive because you don't need all those tensiometer everywhere. However, it's not as efficient as tensiometer. Beside that, even if it's not as efficient, we think you're going to likely reduce significantly water consumption in most nursery. And we saw that with last year where we had tensiometer in just the commercial nursery, and we saw that the soil was staying really, really moist most of the season. So we're pretty sure we're going to be able to reduce the amount of water used with that. The coefficient must be adjusted often during the season, and that's probably the challenge with evapotranspiration. So what's to come? We're going to try to refine evapotranspiration automation next year, and then compare the efficiency of tensiometer and evapotranspiration with other species. So with that, I would like to thank all our partners. So COHA and Agriculture Canada and Fafard Scots, which Fafard is a division of Scots, and Multiplan, which provided us the plant, and Orto with all the automation equipment and tensiometer. So with that, I will be able to take a question if you have.